Thank you. I'd like to thank the organizers for having me. And this is joint work with my former postdoc, Anthony Joseph, who is now at Walmart Research Lab. So since this workshop is from theory to practice, I definitely have a lot of practice, and I'm conjecture that I'm going the furthest to practice for the whole workshop. And you guys can tell me whether it's the case by Friday. So I will motivate what we do with like clustering with a project with a bunch of people, maybe all my student Siji Wu and the scientist at Berkeley Drosophila Genome Project, and Irvin with a collaborator, and Hamans and Sue Saniker. So I think this slide probably is not necessary for this workshop. So Drosophila, for a good reason, has been one of the model organisms for biology to study, and has very small, short life cycle, and the genetic mechanism, at least at the beginning level, very similar to humans, and it's very easy to maintain in the lab. So has been used a lot. And the IBL, Sue Saniker's lab, has been collecting relatively large image database for embryos of Drosophila. And what you see here is you can think this as gene expression intensities of this gene called tailless on this embryo. And tailless means that if you kind of knock out this gene, the fly won't have tail. So the name is quite telling. Okay, so this is a very unique data set in the sense that you have the spatial distribution or layout of the gene expressions relative to the embryo. And most of the other genomic data set have, like RNA-seq, doesn't have this spatial information. So the idea is to use this co-occurrence of spatial information, try to figure out which gene interacts with which. And we have 7,000 genes with, this doesn't work, 1,000 stain embryo because we have replicates, and you can see that they have different shapes. And this is pretty early stage. And the goal is to understand gene interactions and localized, and try to understand how organs are formed.
Sorry,
so this is...
Sorry, where was the data? Is the data set available?
Yes, it's an open data set. It's a public domain, and fortunately or unfortunately, there haven't been too many papers. They would love to have more people use it. So if you're interested, email me. I'll be happy to point you to where the database is. So this is a known kind of a clustering or segregation, segmentation of the embryo based on some very old technology of laser ablation. So you basically try to cut with laser and see what happens. And you can see that for early development, it's mainly about food and skin and a bit neuron development. So that's what happens when you first have the embryos. You try to make sure you can process the food, and you have the skin, the shelter, and then you have neuron, some intelligence. So one test is, this is called the fate map. So with this co-occurrence of different gene expressions, can we recover something like this? And so first we use these gene expression images to form a graph. So let's think that we can register and line up all the images. That's also non-trivial, and become pixels. They all lined up, those stacked up. So for each pixel, you think you have a whole vector of the intensity of different gene expressions at that location. Okay, that's the signature of that location. Okay, so the nodes will be the pixels. And we're going to put an edge between the two nodes or pixels if there are a lot of co-expressions of genes. That means they seem to be related because a lot of transcription or genes are working together at that two locations simultaneously. So we're making assumption, basic co-occurrence means interaction, which might not be the case. But this is, for this data, a necessary assumption. Question, after that, can we recover this map? So we have, that's what I said, right? You have all these gene expressions, and we, after some normalization and centering, we just look at the inner product, and we basically cut at the 90th percentile. Above that, we call it 1 connected and 0 otherwise. So the highly co-occurring pixels with high co-occurring gene signatures, we say they're connected. And now if we use that data with 0, 1 as a graph, and we use unregular, just the usual spectral clustering, that's what you end up with. Very skewed. It doesn't look like the fate map at all, right? That will be the clustering coming out if we just give in the number of classes as 8. If you use the regularized version, which I'll tell you, then get something a lot more similar to what you saw in the fate map. Okay, so this just shows you that regularization does help, and then we try to explain why. You can identify the different parts with names. And this is not satisfying at all, right? It's completely skewed. Because the small clusters skew the big clusters. That's what I think people know is the problem. And in general, we have community detection. And for this workshop, early papers, at least you can go back to the Fiedler paper 1973, and there are more recent works from both CS and STAT community. So remember, I have made a 0-1 matrix depending on how co-occurring two pixels share the same gene expressions. And each node for me is a pixel location. And this is just the usual degree matrix D, and you do the normalized version to get the Laplacian. Well, I'm not doing the I minus because the decomposition is the same. So you can have different versions of it, and we're taking this version. And for people who don't know spectral clustering, this is like a 2 slides of tutorial. You have the simulated 0-1 matrix, and you can do permutation to make them clustered. And you take the first and second eigenvector of the Laplacian, and you can see 2 clusters very nicely. They're the same, except now I say there are 2 clusters here, they're just data points. And you do k-means. Okay? And I already said that. Spectral clustering become very popular, and there are many papers, not exhaustive here, to analyze under different conditions. You can assume like manifold learning type of assumptions, and here I take the more stochastic block model and analyze the spectral clustering under the stochastic block model, more like a graph models. And in a 2013 paper by Amini, Chen, Bickel, and Lavina, Peter is actually here, they propose a very simple but effective regularization. You simply just lift the boat on the adjacency matrix. You add a constant matrix. This is just for normalization. It's just a constant matrix. This is the 1-1-1 matrix, and this is normalized regularization parameter. Okay, so just lift the boat for all the adjacency matrix entries, and then pretend that's your data of adjacency and do the same thing for Laplacian. Everything just becomes the same. Okay, so you just add a little constant to the adjacency matrix, and then proceed to do Laplacian. Okay, and then you take the top eigenvectors and you do k-means, and that's the regularized spectral clustering. And there's another version that you only add the constant to the degree matrix, not the adjacency matrix. So in practice, actually, it might be a little better. That's what I hear. I haven't had a lot of experience. Okay, so you have similar analysis in this line of research.
What's the motivation for this?
The motivation, people probably know better what's the motivation, but from what I can tell, you have basically reduced a lot of noise in the adjacency matrix. The small clusters you don't have signature for that. And then you can concentrate on the big communities. Because the small cluster can always has known to be problematic to destroy, as I show you with the fate map, the big clusters. So this way you kind of downplay those small clusters. And the stochastic block model has been mentioned. is the simplest model. I think it's also called the planted partition model. In the CS literature, you assume you have independent edges drawn with different probabilities independent from Bernoulli's. So just a toss. And then the probabilities can be generally different, but usually you need a separation of the probabilities for the blocks. These probabilities cannot be too close. The P's and Q's cannot be too close, otherwise you don't have a block structure. And this is just, again, a sample, and then you can see that things, you see this block type of adjacency matrix.
So,
And I will focus on two clusters.
a sample.
Yes.
Two questions. First of all, when you say performance increases when you add the all ones matrix,
I'm sorry, what?
so you're adding the all ones matrix
Right.
to the adjacency.
Adjacency matrix.
So
Yes.
this is improving the performance as in the result obtained, or also the running time performance.
The running time is the same.
Okay.
Yeah, but
And so is this equivalent to just increasing all edge weights by?
yeah. So you don't have zero weight anymore. You have a small weight. You kind of just add a constant to everybody. Yeah.
So have you also tried on just the, without the thresholding, just on the correlation matrix itself during the clustering?
I think we get probably similar result. But the thing is, it's harder to see if you have a lot of edges when you do that. And we also kind of do a bit denoising by thresholding.
That's
Yeah.
what I was wondering.
Yeah.
Like the denoising effect versus.
I think if you cut not as high as we just get similar result, but if you don't, I think you might have too noise result. And also it's hard to visualize, just too many links. Yeah. So I don't think it should be too sensitive to the 90%, but I think you do want to threshold a bit. So what we want to show you is to give you some insight on why the regularization works by some bounds. So suppose you have two clusters, and this is a simulated stochastic block model with two blocks, and if you do n, just a regular spectral clustering, you can see that you can still have good separation, but the variability of this direction along the second eigenvector direction much bigger than after regularization. Okay, so this is just same thing as any. When we do a lot of regularization in statistics and machine learning, it's really reduced variability, and you can actually show that the centroids of the two actually stay the same. Okay, so the only thing you are introducing is to reduce variability with the same distance, then you gain. But there's a sweet spot, right? If you add too much, a big tau, then everything becomes constant, you lose the signature of the cluster, right? So apparently you don't want to add too much. This is selected with our method, right? So there's a sweet spot. If you don't add much, nothing will change. If you add a lot, you lose the signature of the community, and you want to be in the middle to really reduce the variance, but still retain the distance. And if you do k-means on this, because the same problem you have, you end up with putting this together, because for here, the cross-cluster distance, as in the previous talk, mentioned that it's smaller than within the cluster, so you messed up. And here, you don't have that problem because you tighten them up. And, yes?
For two classes, you wouldn't use k-means.
What?
You would take the threshold if you only have two classes. You would typically do thresholding, right? You wouldn't. I guess, yeah.
Try to compare it. Yeah, you have to fix some after downstream analysis. Yes.
So if you didn't regularize, the trouble would be that some of the rows might have very small di, and the inverse makes it unstable, right?
Yeah.
So instead, you could just cap off the entries of d inverse just as well instead of adding.
Yeah, the other one only to work with the other regularization.
Uh-huh.
There's another line. We just, because we're close to Peter's group, so we end up analyzing this version. You could do the other way too, just to regularize the D in some way. Yeah. I think it's very similar analysis can be done. So here's just some notation. This is just the data version of the regularized spectral clustering, and use V as the matrix, the top two eigenvectors of the sample version of the regularized spectral clustering. And you can similarly define population versions of the same quantity, right? You take the probability matrix instead of the adjacency matrix, and also do the regularization, and you have parallel Laplacian and so forth for the population quantities. And you just recall this is the sample version, and this is the, with the pop, is the population version. Right, so you want to see how they relate to each other. And you can take the centers of the centroids of the eigenvector decomposition of the Laplacian and take a look at that. So this is the projection on the first two eigendirections of the Laplacian from data. And you have the centroid here and here. And you basically looking at how far apart the centroids relative to the within cluster tightness. And this is the quantity. You're looking at the within cluster distance ratio to cross cluster distance. That tells you how well things are separated. And you can show that actually this actually doesn't depend on tau. So from the point of view, looking at the impact of tau, it doesn't make any difference. And the tau can be rewritten as the eigenvector matrices between the sample Laplacian and the population Laplacian. And with the Davis and Kent type of perturbation theory, you can now bound, because this just disappeared, doesn't depend on tau. I'm only interested in the impact of tau become the difference between the Laplacian, between the eigenvectors of Laplacian. This is the spectral norm, and the second eigenvector, the eigenvalue, which measures the, and this eigenvalue decreases with tau, because when tau is large, then there's no signature, you cannot separate. Then you have a bond which is not very useful. And with something a little bit more severe, using the concentration of matrices in Olivia, you can show that there's this more precise bond than the rough bond. You have the eigenvalue for the, from the first cluster and the second cluster. So better bonds can be obtained using some more refined techniques in Blaschnan's et al. Paper from 2011. So if you unpack this result for the two-block stochastic block model, you see that we don't need to put constraint on the minimum degree of expected degree of the nodes. We can get away with the average expected degree of nodes, that you'll be able to recover the clusters. So that's the effect. You push down the small clusters, and for two clusters, you get a big cluster right. Of course, you get a small cluster right. But in general, you're not so lucky. You're able to get the major clusters correctly, not the small clusters. And we don't need a minimum degree assumption. So from the statistical point of view, the most interesting is actually the how do you select the number, the regularization parameter. Right? Now you introduce a smoothing parameter. Are you going to be able to find a data-driven way to do that? So what we did is that we take advantage of the theoretical bound and come up with a data-driven criteria to do the selection. So the idea is that we just look at the quantity which come out in upper bound, which involve tau, which is the regularization parameter. And here, involves a population quantity which we don't have any access to. Instead, we're going to use the model, stochastic block model, or later actually also works, even we don't have the theory worked out, we think it can be done, or degree corrected stochastic block model, to have a parametric model, and then use that to estimate this population version. We have a surrogate and plug in. And then we just minimize this statistic. It's the upper bound, it's the estimated upper bound. And that's how our data-driven regularization parameter chosen. Okay, so it's pretty simple idea using the upper bound. And if you do that, then you see that you do improve. You basically have, using the output from the regularized, for any fixed tau, you have the clusters using the regularized SC, stochastic clustering algorithm. And then with the clustering, you can estimate easily the stochastic block model. Without that, it's kind of hard to estimate stochastic block model. Same time with the clustering, you only have the estimate parameter. So the second stage is pretty easy. And then you use this probability to estimate the population version Laplacian and the eigenvalue. And then so for each tau on a grid, you now have a quantity and you can optimize. And that you end up with another run of the same stochastic block model, things a little tighter, and then you're going to, that's what's chosen by our data-driven method. And again, k-means won't be able to accommodate this kind of overlapping in this direction. And we also messed up a bit with regularized version, but much less. Okay. So it's really reducing the variability. That's what regularization does, as in many other situations.
Okay.
Yes?
Does it make sense to iterate? Iterate. After you do this, do it again.
So iterate which stage? For fixed tau?
No, so you re-estimate the population.
After you have the tau?
Yeah.
We haven't. You could. We haven't, because it's pretty heavy already computation-wise. Because you do many k-means and like a spectral clustering. I think, I don't know whether we'll improve or not, but computation-wise will be even heavier. So for the political block data, so this is from this paper and being used a lot in community detection. People already labeled the blocks as conservative blocks or liberal blocks. And you can see that there are few kind of outliers. And if you use spectral clustering, they will really hide the big clusters, at least for the leading eigenvectors. If you look at the degrees, it's kind of the parallel. And you look at unregularized spectral clustering, the first and the second eigenvector, you don't really see a separation. Okay. Because it's dominated by these outliers.
How did you get the edges?
What?
Where did the edges come from?
Oh, they're just linking to each other. They refer to each other.
Okay.
Yeah.
So is it symmetrized? So
It's symmetrized. We didn't do the direction. Yes, this is symmetrized because I'm not dealing with the direction. So there's a link one way or the other. We just call it a link. So the second vector basically end up concentrating on discriminating this small cluster from the rest. It doesn't really tell you this major cluster. But the third eigenvector does differentiate the blue from the red. So you can also think of the regularization as shifting the order of things and now let you concentrate on something which is hidden in the third eigenvector and lift it up to the second. And if you do the regularized chosen by our method, tau being 2.5, you can see now the second eigenvector already differentiate the two main liberal blocks versus the conservative blocks. And but I should mention that one of the authors of the regularization paper, Yu Chen, in personal communication told us he also tried using the modularity, Gervin-Newman modularity, and used that. It works pretty well too. But it was not, they didn't put it in the paper. So that's another alternative method to do this. We did compare, if you simulate from stochastic block model, our method works better, but we only try 102 data set, the results are pretty comparable. So if you have really like stochastic block structure, because our model really assumes estimator stochastic block, we have an edge. But in terms of when you don't have that, something like stochastic block model in your data, then with the limited experience we have, the other idea from IE also works pretty well. You're just looking at the Gervin-Newman modularity and then compare that for different types. So we have that in the paper. I think we did a comparison in the paper. So back to the fate map. You can see that this unregularized spectral clustering is being pulled like in the political block model, that you have these tiny kind of clusters that dominated the leading eigenvectors and skewed the big clusters. And if you regularize, you get the more sensible segmentation of the embryo, which much better with the fate map than this one. And the summary is that you can interpret upper bound as something like bias and variance trade-off. The difference, remember the bound you saw a couple times, also the motivation for regularization, parameter selection. The difference between the the spectral distance between the sample Laplacian and the population Laplacian is like variance. And you have downstairs the eigenvector, which is like, eigenvalue is like a bias. And if you take a log of that, you have like more like log variance plus log bias. So there's that trade-off, which you see a lot in regularization literature. It's always playing this trade-off. So it's kind of there too in a different form than what we used to L2, like just bias square plus variance square, variance. And we find our threat-to analysis have a practical implication in the sense that we can select the regularization parameter. Even we didn't do the theory, we believe it can be done. We also tried to use degree-corrected SDBM in the estimated population Laplacian, and it works better because it's more flexible in practice. And for fruit fly image segmentation, it seems promising. And the paper, we have a revision 2014 at this website. And what's going on for the project is that we want to go to the later stage for the embryo to find out where are the organs. And for spectral clustering, as I mentioned to Jerry, that we need fast algorithms. Now it seems quite heavy computationally. And we think that the regularization has a nice correspondence with this level set idea started from Hartigan's work. It's really kind of giving you a level for what's the level of cluster you want to look at. And for continuous data, I think it would be interesting to get the theory done for the regularization parameter selection. Thank you. So I think I'm getting you guys good lunch.
In the gene expression data, is the time of development the same for all the images, or different times of development? And
that's actually, we so far use human manually matching the stages. Right now, it would be great to have temporal because it's development, right? But right now, this is high throughput data. We have the timestamp is very coarse. There's some better data for fewer gene expression from Michael Biggins' lab, which is more continuous. But for the high throughput with 700 genes, we only have very rough type of, and then it's eyeballing. We kind of humanly put them together. And that's another thing. If we can do segmentation better, that can also help us to line them up better. So right now we're assuming they're lined up, but later you want to have a kind of inner loop, right? Use the organ development stage, maybe help us to line up. Yeah. We're not there yet. Yeah.
So it seems like the graphs must be relatively small before you can do that, because that would blow up your memory. But, you know, putting in the complete graph. So you must do some, either the examples are small, but the other way, of course, would be to put in an expander or a random graph of low weight. But then I think there was a bunch of papers that did that, I think, in image processing. I think some of Malik students did that.
Which student? Malik, okay.
During the Malik. I thought they added random graph connections.
For the large graphs.
Similar to yours, right?
Yeah, for me, my graph is relatively small. I have like a few hundred pixels.
So you're adding in all edges, right? But I think theoretically, you're adding in all pairs of edges, right?
No, but that's thresholded, so it's pretty sparse.
Yeah, but still, it's still all edges. It's not sparse. It's still dense graph theoretically.
No, but because I have correlation, I cut it at 90 percentile. The edges below 90%, I set it to 0.
Are they sampled with those probabilities?
What?
The edges are actually sampled with those probabilities?
When we didn't sample, we just cut it, right? So you calculate all pairwise correlations, and then you cut at 90th percentile, and the ones below 90%, we just set it to 0.
But you send this off to something to find the Fiedler vector for you, right? At some point, you need to compute an eigenvector?
Yeah.
So that's done on with the regularizer added?
Yes. Then you dance again, you're right. Yeah. Yeah.
And so what I was proposing is to use a random graph instead, right?
So you will randomly select the edges?
Yeah, just randomly put in edges. Because
Okay.
theoretically that should have the same effect.
Okay. Yeah, we haven't tried. Maybe that's how we speed it up.
Right. But then I think there's older work that did that. And
So
I was
Malik's.
recommending some of Malik's students.
Okay.
did that.
Well, okay, I will look. Do you remember when was that?
Yeah, I don't know.
Right after they did the spectral clustering paper?
Basically part of that. I think she was at CMU at the time. I forgot her name. But.
Okay, I'll chase after that. Thanks.
Sorry, but if computation is an issue, I mean, these eigenvectors are computed iteratively. So, at every iteration, you only need to compute the A times a vector. And it's a rank 1 perturbation, so it's still order n. You never construct the full matrix. You just do A times V plus 1 transpose V.
Yeah, but if this is billion sizes, then that's not going to do very well. That's not, I mean, all the amount of stuff we always do is inverse powering.
Yeah, no, that would
Inverse powering is much
None of
faster.
you would do 4 powering. You lose 1 order of convergence, but then it's order n. And you don't have the problem of constructing the full matrix.
But then you have to do some multiple event iterations, right?
That's correct. Well, it depends on the gap, no multiple event.
But if you do inverse powering, then you only do log iteration.
Well, here it depends on the gap, right? So the regularization, it depends. If the gap becomes larger, actually you might
Yeah, definitely follow up with you guys. The computation, we just used pretty basic, like R type of thing. So, not.
How big are you?
For me, it's just a few hundred. Yeah, because the pixels, originally it's much higher precision, but we had super pixels. But eventually, we like to do higher, right? But so I'd love to talk more about that. Yeah.
When selecting tau, you should estimate the second eigenvalue of that Laplacian. So it is possible that this eigenvalue is negative. Like in 2 blocks. So you calculate Laplacian, which is a 2
It's a
by
symmetric
block matrix, right? So here,
it's symmetric. Oh, you mean the estimated one?
yeah. Since the first eigenvalue should be positive, but the second one is not guaranteed to be positive. So
You mean the estimated version?
yeah, estimated version. So when it is negative,
We'll probably just. I think it hasn't happened in real data with us. But in theory, you're right, because he's worried that the estimate stochastic block model might not be symmetric. No.
but even
Yeah, you worry about if it's symmetric, then you have positive eigenvalue. Because I symmetrized the.
Yeah, but the estimate version of that Laplacian is not guaranteed to be positive semi-definite, right?
When you plug in the parameter estimates.
Always positives.
Yeah. Okay.
Any other questions?
Wonder.
So in all your examples, the two clusters that you're dividing into, they seem to be comparable in size. So in some other instances of clustering, sometimes you want to cluster a fairly small cluster to separate a fairly small set from a fairly large set.
Well,
Does that regularization change when you?
so I think that's what I think what intuitively I think what the regularization does. If you look at the fate map, they're not always the same size. So in some sense, you kind of cut it at somewhere, only go for the ones above that threshold.
So I'm a little worried that if your one cluster is much smaller than the other, all of a sudden you introduce a lot more.
We just don't, we push the small cluster down and we don't see them. So you're right, that would be a problem if you really want to go for the small clusters. Right, then the regularization will obscure that.
Are there other methods of regularization that might be promising for dealing with that?
Well, the thing is the chicken egg thing, right? For clustering anywhere, if it's very small, is there noise or is there not? It's not very clear. Unless you have prior information to tell you that's a real cluster, it might just be noise, right? So I think other prior information have to come in.
So if you just add a perturbation on the D rather than on the whole.
Yeah, that's what the other line,
Just probably are the same.
quite similar. But what I hear from that group is that it works better, but I don't have first-hand experience.
Yeah.
They think it's better than doing both.
Sparsity.
That's what the same,
Yeah.
it's similar, you just regulate what's on the bottom. I think the analysis Carl Rowe's group did that. It's quite similar. I don't think the theoretical analysis would tell you it's better, but in practice, his student tells me that it's a little better doing that way. But we haven't tried it.
Our experience is opposite.
Is the opposite?
Yeah. It's yours better.
No, I mean the one from Peter's group. Yeah. So then maybe it depends then. Well, we haven't compared. I think for our data, I think it's quite similar. Yeah. But I did hear from the other group they think it's better, but I haven't had first-hand experience. So maybe, yeah, I'll put you in touch with them and you guys can figure it out. One.
We use different examples.
Yeah, exactly. Yeah. But to figure out when and why one is better than the other. But I don't think that asymptotic analysis really tell you that. It's probably coarse, not detailed enough.
Thank you.
Thank you.